Good morning, class 11. Now today I will continue the same chapter, cell cycle, but the next part of it, that is cell division by meiosis. So today I will discuss about the meiosis. First I want to tell you what are the significances of mitosis, or you can say why mitosis is important. First and foremost, mitosis is very essential, or it is a vital process for our growth. Next one, for our repairing our body tissues. Third, it helps to surface volume ratio of the cell. This is the children, it is the new for you. Surface volume ratio. It means it restricts the growth of the cell according to the size. According to the size means all the cells are not being in a proper size according to the person's height, according to the person's length, according to the person's muscles. So width, that is width, the cell will increase in their surface volume ratio and number of cells will be viable or variable during their lifetime. Next is nucleoplasmic index. This is also the size of the nucleus and amount of the cytoplasm. These two should be in proper ratio. That is called as nucleoplasmic index. It never can be happen that cell is bigger in size, so there will be more amount of cytoplasm and nucleus is smaller according to it. So this is maintained by the mitosis. So I am telling you that main 4 significances are there for mitosis. First, that is growth. After growth, there will be the development of the organ. Then third is repairing of the tissues. Maybe it is destroyed for any reason. If it has or if it can be repaired, then can be repaired by the mitosis. And next one, they can maintain the nucleoplasmic index, that means amount of the cytoplasm and size of the nucleus remain in the proper ratio throughout the life. And next is surface volume ratio. The outer, that is cell membrane, it forms the surface, and according to the surface, the volume of the cell should be how much. It can be maintained also by the mitosis. So these are the significances of mitosis. Now I am coming to the next cell division, meiosis. So meiosis, see, in short I am telling you here, suppose this is a diploid cell, that is 2n number of chromosome it is having. Now when it starts meiosis, first division is called as reduction division. Reduction means the number of chromosome becomes reduced. So the name of the cell division is given here, reduction division. Now reduction division is also called as first meiotic division. That means first part of the meiosis cell division is called as reduction division. And here the number of chromosomes become, which was diploid, now they become haploid. So one diploid cell will give 2 haploid cells after reduction division. Now I am coming to the reduction division. In the reduction division, it is also having all the same names, that is prophase, then metaphase, third one is anaphase, and fourth one is telophase. these are having the same name, but this is the first meiotic division. So everywhere you should have to put part 1, metaphase 1, prophase 1, anaphase 1 and telophase 1. So all these 4 phases belongs to the first meiotic division. Now children, first meiotic division, or when we will start the first stage, that is the prophase 1. Prophase 1 is divided into 5 phases. First is leptotene, second is zygotene, third is pachytene, this is pachytene, fourth is diplotene and fifth is diakinesis. All these 5 subphases will form the prophase 1. Now leptotene, within the meaning of it, you can understand about the phases, and altogether 5 phases forms a long period, but with one phase only will form the short period. Now leptotene, leptos means slender, nema means thread-like. Here the chromosomes are thin, long, thread-like in the nucleus in the form of chromonema, not in the form of chromatids. This is the first condition, that is leptotene. Now, in case of mitosis, you learned about that. In the prophase stage, all the nuclear membrane start to disappear. Nucleolus also disappearing, starting to disappear. In this way, here also will be the same. This is the nucleolus. This is the nuclear membrane. Here I draw here with the dotted line, that means it starts to disappear. Now this is the leptotene. In that leptotene, all the chromonema and the half of the total chromosome, all the chromonema, those who are present there, may be any number. Half of them will be the parental and half of them will be from the maternal side. And when they becomes homologous, or they, children, you know about the homologous chromosome, those who have centromere in their proper place, or they have same length of their size. So homologous chromosome come closer to each other and then they start pairing. So then come to this second stage. This is zygotene. Zygos means joining. Zygos, or the meaning of the zygos is joining. Here the parental and maternal chromosomes come closer, but should be the homologous. Homologous pairs are formed in the zygotene, and while that homologous or the pairing, they become procentric. Procentric means the pairing they start from their centrosome. Centromere. Pairing they will start from the centromere. Then next is pro-terminal. Terminal, as the name suggests, they start from the any term or any end. That is pro-terminal way. And intermediate means in between. In between any place they start the pairing and then finally they paired, and here because of that pairing, this process is also called as, or the process is also happened, that is synapses, and this synapses forms them bivalent, because now one chromosome become 2 or they become in pair. This is the zygotene. After zygotene there is now children, here the the centrosome starts dividing or starts going to the another pole. Here both were on the same pole. Now in the zygotene they start to go from the one pole to another pole. One centrosome will be there, another one will be on the next pole. Then in the pachytene, both the centrosome, here they remain in two different poles, and this is the nucleolus. Nucleolus becomes thin. Thin means not that means thickness becomes less because of their disappearance. And now all the chromosomes, those who were bivalent, now they become tetravalent, or they get crossed with each other, and in this way there is another phenomenon known as crossing over. So children, crossing over. See children, during crossing over, this is the centromere, these two are the parts, and now they are bivalent. So crossing over during their homologous pairing, so non-sister chromatids or non-sister parts of the chromosome gets crossed with each other. So this point, this is the point, and this is the point. This is the, just like your crossing on road, and this point is known as synaptonyma, or the complex is called as synaptonymal complex, and this point is called as the synapsis. And after this pachytene, they start their crossing over. This is called as crossing over. Now now children, you should understand what is the sister chromatid and what is the non-sister chromatid. See, if the sister chromatids, these two will be crossed with each other, then same character will pass on from each to one to everyone, then there is no change or no variation takes place. Now one is paternal, one is maternal chromosome, so they start crossing over, that is non-sister chromatid. One is black and one is red. Here I draw here. So one chromatid of black and one chromatid of red, they crossed over each other, and at the joining place there is the mixing of synapses, that is the material, it is called as synaptic complex. Due to that, in the next generation, children, now the chromosome will be like that. The black colored chromosome having some characters from the red colored chromosome, and the red colored chromosome, they will have some characters from the black colored chromosome. So this is the process of crossing over, and this crossing over starts taking place in pachytene. Here, see, they started their crossing over process. And due to that crossing over, only by meiosis the characters can be changed or can be passed from parents to offspring, either from male to female or from female to male, anywhere, elsewhere. So this is the process of crossing over, takes place in pachytene. Now children, after pachytene there will be the diplotin. Diplos means double. Here they become completed their process crossing over, and they separated out or becomes separated from each other according to their proper characteristics. This is the diplotin. And next one is diakinesis. In the diakinesis, see, all the chromosomes gets separated, total nuclear membrane is lost, and nucleolus also become lost or it is destroyed. Now according to 4 chromosome only up to last, there is also the 4, there will be also the 4 chromosome, but all they have some different characters according to their crossing over, after their crossing over. And now these crossed over chromosomes have parental character also and maternal character also, paternal and maternal both. So these are 5 sub-stages of prophase. First is leptotin, then leptotin, zygotin, pachytin, then diplotin, and then dikinesis. So according to these 5 sub-stages, they can complete the prophase 1 only, not other phases, only prophase 1. So altogether prophase 1, it is very important for the crossing over in the meiosis, and crossing over, this is the point, that is crossing point is called as chiasma, and formation of this chiasma is called as chiasmata formation. Now if the chiasma will not form, then paternal and maternal characters will not be mixed together, then there will be no variation, and if there will be no variation, then there will be very problematic condition we will get about our evolution. So first, the chiasma or function of chiasma, they hold the homologous chromosome in their proper place. According to the chiasma, they are sticked along with each other, they never can be separated. And second thing, they can exchange their genetic material to bring the variation, then evolution, or helps to carry the evolution throughout our life. That's why human nature is there. Every time they can think about the new varieties, new thing, they can learn it and they can understand about it only due to the formation of that chiasma, or you can say the formation of crossing over. So we have this type of effect in human body. Not only human body, those who have sexual reproduction, any living organism, if they have sexual reproduction, then they have meiosis, and then after meiosis they can get the mixed character and they are very easily ready to go for evolution. One point I want to tell you, meiosis is very important cell division, because if there will be the combination of male gamete and female gamete, the number of chromosome in each and every time it gets doubled. If the number of chromosome will be changed, then the species also will be changed, and the species if changed, then the variety of the animals, you can say, or a variety of the living things, living organisms also be changed. So one race never can be alive for the long time. Simply they will live for one generation. So it never happens. That's why meiosis can reduce the number of chromosome in the cell division, and after that one there will be the male gamete and one there will be the female gamete. When they will combine, again they will form the diploid cell, and same race can be alive for the next generation for the long time period. So for the today, this is over. Thank you, children. These are the five subphases for the prophase. Now tomorrow or in the next video I will continue about the other phases. Thank you, children. God bless.